Hello friends, welcome to PV Breakdowns. From our previous lesson on DNA structure part 1, I introduced what DNA is and described the basic components of the DNA structure. Now let's discuss some other basic features of the DNA structure. The DNA double helix consists of two types of grooves, namely the major groove and minor groove. For knowledge sake, a groove is just like a narrow cut or depression in a material. And one thing about a groove is that you can fit something into it. For example, this is a wood groove and I can fit this pen into it. You can also see grooves on the surface of motor tires. Now back to DNA grooves. The major groove and minor groove alternate. That is, they occur one after the other. So you have minor groove, major groove, minor groove, major groove, minor groove, and it continues like that. And both grooves run continuously along the entire length of the DNA double helix. Also, the major groove, as you can see, is wider and deeper than the minor groove. Now, if you must know, the DNA grooves are very important, and this is because they expose stretches of DNA sequence. You know, these base pairs form a sequence like a continuous series. And as a result of this exposure, regulatory proteins called transcription factors are able to recognize and bind to specific DNA sequences. Again, if you must know, transcription factors are DNA binding proteins that control the rate of transcription of genes. Their work is to simply turn on and turn off genes. Remember, I said if you must know. Now, lastly, let's talk about the measurements and or dimensions of the DNA double helix. On average, a DNA molecule, when fully stretched, reaches up to 2 inches long, and 2 inches, when converted to meters, gives us 0.0508 meters. Now, when describing the measurement or dimensions of the DNA double helix, we typically use a unit called angstrom. Angstrom. And this is the symbol for angstrom. So 1 angstrom is equal to 10 raised to the power of minus 10 meters, or 0.1 nanometers. Now, the length of one complete helical turn in the DNA molecule is up to 36 angstrom. So this means that from this point to this point makes one complete helical turn, which measures up to 36 angstrom, and from this point to another point makes another complete helical turn, which measures up to 36 angstrom as well. And there are 10.5 base pairs in each helical turn. This means that there are 10.5 base pairs from this point to this point, although the number here might not be up to 10.5, but the standard is 10.5. Also, each helical turn, each complete helical turn has a major groove and a minor groove. Now, the distance between adjacent or neighboring stacked base pairs is about 3.4 angstrom, and it is important that this distance remains constant so as to maintain the van der Waals attraction between the stacked bases. Finally, the diameter of the DNA double helix is about 20 angstrom. So if you enjoyed this lesson, it should be in your DNA to click the like button subscribe to my YouTube channel, and share with your friends. And don't forget to do the right thing and choose wisely.